Okay, assalamu alaikum, and welcome, and Masa Al here, and I hope that you're all keeping well. I'm Cormac Gahan, and I will be giving this lecture today on. Oops. Go back to the start. Yes, I'll be giving this lecture today on techniques used in biotechnology, and also some of these are also used as diagnostic tests. So we're talking about the future tests really in personalized pharmacy, and some of these are mentioned here. And so we get straight into it because we've got quite a lot to cover today, and I realize there's quite a lot of detail, but I hope that you'll be able to go back and go through the slides and understand the detail when you go back over it. So don't be overwhelmed today if you think there's too much. You'll be able to go back, and you should be able to follow the slides. So what we're talking about today are techniques that are used to investigate all the aspects of the information that are contained in a cell. So this could be DNA. So what are the techniques that we use to understand the DNA molecule in the cell, the sequence of the DNA, or its presence? What about if we want to see, well, what genes is a cell expressing? What RNA is it expressing? Well, we can use techniques for that as well. This is called transcriptomics. And then proteomics describes the protein. So this is all the aspects of information in the cell can be analyzed. So the first thing we'll start off with is genomics. How do we understand whether a gene is present or what the sequence of that gene is? Okay. And so this examines genetic sequence information. And the first actual test is called a Southern blot. And a Southern blot is used to tell us whether a gene of interest is present. So is that, or a piece of a gene of interest? So is that piece of a gene present in a sample? And it's quite a simple technique created many, many years ago. And what it does is you run your sample. So this could be a sample of interest, biological sample. You run it on a gel. So you're running the DNA on a gel, all of the DNA. Usually you would chop up the DNA using a restriction enzyme, chop it up, run it on a gel, but you can actually transfer that DNA to a nitrocellulose filter. So you press it onto a filter, and this actually means that it's a bit more stable. So it's transferred to a filter, then you add a probe, and this is a probe to the gene of interest. So you can see this is a probe in red, and it's binding to the gene that you want to find. Okay, it's complementary. So you want to see, is this gene present? Use a probe to that gene. The probe is bound to a radiolabel, so it's called a radiolabeled probe, which is radioactive. And wherever you use it to hybridize to a complementary DNA sequence, and wherever that piece of DNA is present, the probe will bind. Okay, so here you can see it's actually binding in 2 places, and so on. So this, even though it looks simple, it gives you a lot of information. It tells you, well, that gene is present, and maybe it's present twice here, or maybe this gene has been chopped up in the middle by a restriction enzyme, so it can give you a bit of a fingerprint of the DNA sample. Okay, so it's a very straightforward, that's our first technique, very straightforward technique to look at whether a gene is present. Now, what about if you want to find out a bit more about the sequence of that gene? And you need to use a thing called DNA sequencing. And what I'm going to give you over the next 5 or 6 slides is a history of DNA sequencing. So how do we know G, C, A, T? How do we know that sequence in a sample, in a DNA sample? And can we even understand the entire genome sequence of humans and others? And so this all goes back to the 1970s, when there were 2 different techniques developed, and we're just going to look at the chain terminating technique. So I'm just going to focus on one of these. This is the most widely used, developed by Fred Sanger. He got the Nobel Prize for his technique. Okay, there's quite a bit of writing on this on the screen, and you can read through this later as just for your notes. But the most important piece here is that chain termination, it is a technique that uses a DNA polymerase to make a copy of the DNA that you want to sequence. So you want to sequence this gene, but you use a DNA polymerase to make a copy. And it's that copy which actually gives you the sequence. Okay. And so it's very like PCR, which you've done in a few different lectures. You have a DNA polymerase, which makes a copy, and it incorporates, in making the copy, it incorporates these nucleotides. Okay, so that's fairly straightforward. But the big difference is that you can also add these chain terminators. And these are dideoxynucleotides, DD, ATP, and so on. And these are chain terminators. And what they do is they stop the reaction. Okay, so this will be clear on the next slide. So the thing is that these are added at very low amounts. Okay, so these are added at low amounts, whereas these are added at higher amounts. And by adding them at lower amounts, it means that they're not always incorporated. Okay, they're not always incorporated. So what is done in practice, and what used to be done in the 1970s, was that if you wanted to sequence a piece of DNA, you take that DNA and you'd add it to 4 different tubes. You'd add the DNA polymerase, you'd add the nucleotides, and to each individual tube, you would add either DD-GTP, so the chain terminator for G. In the next tube, you'd add the chain terminator for C, T, or A, and so on. So you'd be carrying out 4 separate reactions. And so if we take the reaction for G here, well, the chain terminator for G is added at very low amounts. So some of the samples will give some of the copies will give you this big long copy here, where there's just one chain terminator added right at the end, which stops the reaction. You're also going to get in this mixture, you're going to get all of these mixtures. You'll also find this one present here, where it stops here. Okay, so by random chance, it has incorporated the terminator here. That gives you that size molecule. Here's another one here, another one here, and here. So wherever there's a G, it means you can get termination in that of the sequence in that sample. Okay, so if we just take the G sample, so if we just take this tube, we can run all these on a gel, and what you'll find is that, by the way, in blue here that's just the primer. That's the primer that's used. We're really interested in this black sequence here, okay, with the G. Okay, so this particular piece of DNA runs furthest on the gel, and this is the smallest one, this guy up here. Okay, and whereas the largest piece doesn't run very far on the gel, and this is the largest one here, this one here. Okay, so what this is giving you is a bit of information. It's giving you information on where the G's are in the sample. But it's not until you put all the information together that you know the sequence in that sample. Okay, because this is the information for G. Okay, so you've got all of these pieces. You've got this short piece here, longer piece can occur, and so on. For the C's, it's different. You can get a very short piece here, T to C, or you can get a longer piece, this T all the way down to here, and all the bonds in between. This is for T and this is for A. And it's putting all this information together that allows us to piece together the sequence of the gene. Okay, and this is actually on the next slide. This is exactly how these experiments used to be run, and this is just giving you the basic background to gene sequencing that was carried out in the 1970s. And what they would do is they'd set up all these reactions. 4 reactions would be set up, and then you'd actually run them, the G reaction, A, T, and C, all in separate gels. You put all the gels together, and then you could read them like this. So the T is the first one here, then C, C, because these all end in C. This one ends in T, this one ends in A, and A. So it's telling you where these all end. So you're working back T, C, C, T. So T, C, C, T is the readout. Okay, so you can work your way up this by reading the sequence in in these different lanes. Okay, so the shortest one that runs furthest is the T. So this makes sense because here's the T here. Okay, and then another short one here is C or C, and so on. So hopefully, look, I know you might need to go back over this and look at this in a bit more detail, but that is the nature of the early days of sequencing. So the very first sequencing method. And this was revolutionized by another researcher called Leroy Hood, who developed a much quicker method and more mechanized method where you can use a machine to do the same method. And look, you can read, again, I'm just saying that this is directed reading. You can read through this yourself. But the most important thing is that he developed fluorescent dyes of different colors, one for each of the 4 DNA bases. Okay, so in other words, he made, and that's in the next slide, he made the DDA green, the C is blue, the G is yellow, and the T is red. Okay, so he made different colored dyes for the chain terminators. So now he could run all of this sample in one tube, because it meant that each piece of DNA, it now ends in yellow for G. So this small piece here ends in yellow for G. This that's the next size up, ends in blue, which is C, C, G, and so on. Okay, so now he could produce all of these gels. This is all big gels here, and all with different colors showing you where each piece ends. Okay, now humans can't really read that, but a machine can read this quite easily, and it will tell you exactly where each piece ends. And this gives you a readout of the DNA. Okay, so hopefully you'll see how we can go from this. We could go from this scenario to this, which means you can run them all in one tube, and a machine can do it to generate lots and lots of different readouts. Okay, lots of information. So what that technology did was, that automated technology did, was it led to the Human Genome Project. And the Human Genome Project was the challenge to sequence the entire human genome. All of the chromosomes, all of the DNA was to be sequenced from a single individual. And this was not so long ago, really, when you think about it. It was only in 1990, not too long ago. And they gave themselves 15 years. It was actually completed in 2003. So also not very long ago, really. And the aim was to identify all of the genes, to identify all of the nucleotides sequence. But of course, it raised some ethical issues. For instance, some companies got together, like Tiger, and they said, well, we're gonna make a company, we're gonna get investors to invest, and we're gonna beat you, we're gonna actually generate this information more quickly. Okay, and then we're going to own this information. So who owns the information, and how can we develop drugs, and they can develop drugs, and so on. So this led to some ethical problems, but they sorted it all out, and all of these companies got together. They got together with the universities, and they said, we're going to do this together. And actually, in 2001, they published the draft genomes, and it led to some really interesting findings, because they found that, well, for a start, very little of the DNA actually codes for proteins. We don't know what the rest of the DNA does. Okay, what does it all do? Very relatively little. It's probably involved in replication and regulation of gene expression. They only found 20,000 genes. We said this in the previous lecture. Only around 20,000 to maybe 30,000 genes. Well, before that, it was thought that we had 200,000 genes. So a big difference. And this means that much of the genome is involved in gene regulation, and the complexity of humans comes from gene regulation. We talked about that the last day. Most is unknown function, but they did isolate and found genes associated with breast cancer, muscle disease, deafness, blindness. Lots of different genes have been identified that play a role in human health. And this is really important for pharmacy students, because we've been able to map 3 million single nucleotide polymorphisms, which are areas where genes differ where my gene would differ from the vast majority. Okay, and what this means is that I would have a mutation in a gene, and that might influence my appearance, or it might influence my immune system. It might influence my susceptibility to diseases, muscle diseases, to Alzheimer's disease, to Crohn's disease, and so on. So this is really where the area is going. What makes us different? And to understand what makes individuals different, you have to sequence individual genomes. And in England, in the UK, they developed the 100,000 Genome Project, which is currently underway. Sequencing has been completed for about nearly most of these, I think. So in 2018, it was they sequenced 50,000 human genomes. They want to get the number up to 100,000 human genomes. And this will allow us to map SNPs and so on. Okay, single nucleotide polymorphisms. Now, just to bring you a bit more up to date with sequencing technologies, all of this is being facilitated by much more rapid sequencing technologies. We don't just rely on the old-fashioned technology that I've outlined already. There are always new technologies coming along that are speeding things up. And this has led to next-generation sequencing. And in next-generation sequencing, what you do is you have your genome, you take your human genome, okay? You can fragment it, you can chop it up into small pieces. And then each of these small pieces is bound to an adapter, okay? And you'll see what an adapter is at the moment. So it'll be bound to an adapter here and here. And what these adapters do are a couple of things. One is the blue adapters allow the piece of DNA to stick to, they allow the DNA to stick to a board, like a slide. Okay, so the DNA sticks to the slide. This red adapter sticks on here. And what this allows is then for us to add a primer, which will recognize all of these adapters, same primer, okay? Because for DNA synthesis, you need a primer. So this primer will bind to the red part, and then you can start adding your nucleotides, which are different colors, okay? So if you add, say, the yellow one, maybe that's a G, then if that's added, then you know that there's a G here, which is complementary to the C. So it's like PCR, the process of PCR, but it's actually creating just a single fragment here that's complementary here, okay? So these these start to build a picture of the DNA sequence. Okay, so there's obviously DNA polymerase here, and it's allowing that to occur. Okay, so as I said, it's a very complicated system. We don't really need to know the details, except that what you're going to see is that it's multiply high throughput. Okay, in other words, using one of these machines, you can sequence lots of different DNA all at the same time. Okay, we're not limited to reading one piece of DNA, but you're reading lots of pieces of DNA, all in these colonies here. And it looks a bit something like this, where the signal looks like a mess of colors, but the computer can read the signal, and it can tell when a G has been added to one of these dots, or when a T has been added to another of these dots, and so on. So it's tracking all of these dots. It's building up a picture of the gene sequence for each dot. Okay, so it's really a way of, it's called parallel sequencing, where at the same time you're sequencing lots of different pieces. And you can see that this speeds things up, and it actually drives down the cost. So, so what I think you're going to gather is that there's lots of different types of next generation sequencing. Some of them use very different techniques, droplets and so on. We certainly don't have time. There's no need to go into the details. What it's telling you is that the technology is improving so quickly that I think very soon we'll be able to very rapidly sequence a DNA sample from a patient, and to be able to tell you almost immediately what is wrong with that patient. So we're not too far away from that. As I said, for the time being, SNP mapping is the most important thing, where we can tell, well, is it, the Alzheimer's gene? There's a couple of genes that may be involved in Alzheimer's, or maybe there's certainly genes that are involved in Crohn's disease. Can we tell if a patient is likely to develop Crohn's disease? We can, because we can PCR up the gene, and we can use these SNP chips to determine whether they hybridize to the chip. Okay, you can tell. So this is just a very quick method to tell whether a patient has a mutation in a particular gene, and you can map these using these technologies. Okay, here's a recent interesting example from the COVID era. This paper came out last year. What they were doing was they were analyzing a patient with severe COVID. This patient had developed severe coronavirus infection just last year, and they actually sequenced the DNA from the patient, and they found that there was a known SNP that's associated with increased susceptibility to influenza in the immune system of this person. So they could tell from looking at the SNPs in the immune system that this person had a very low antiviral immune response. The production of interferons was poor, and this was actually discovered by the 1000 Genomes Project, if you like, so a few years back. And so more work is necessary, but it certainly shows that in certain cases of severe coronavirus infection, maybe the patient has a mutation in the immune system genes that prevents them from responding properly. Okay, so I just put this as an example. If we can understand this, then we can understand what's happening in a patient. And I think really this is the future of pharmacy, genomics and medicine. Finding out whether a patient is potentially going to develop a rare disease. We can sequence microbial pathogens, bacterial pathogens, sequence the coronavirus very quickly. We can sequence whether they're susceptible to cancer, and we can sequence the cancer itself. Okay, so molecular oncology. And we're going to give a couple of examples of that in a moment. Okay, so in this lecture, in other words, can you take the tumor from a patient and examine that for mutations, gene expression, and so on. And then you give the patient the right medicine for that tumor. So it's called personalized medicine, probably the future of pharmacy. A really interesting thing is also we can analyze how people metabolize drugs. By looking at the DNA, you can tell if they have mutations in liver enzymes that are involved in drug metabolism. If they have a mutation in a liver enzyme, maybe they don't metabolize drugs very well, maybe they're prone to drug toxicity, and so on. Okay. So that's looking at the DNA. Well, what if we want to look at transcriptome? In other words, we want to see what genes are being expressed in a sample. So in a cell from a patient, what genes are being expressed? We saw in my lecture, not all genes are switched on at any time. Not all genes are always switched on. They respond. So can we use techniques called transcriptomics to see if RNA is being expressed in a sample? So what's the RNA situation in a sample? Well, you can use blotting, which is similar to the southern blot. This is called a northern blot, where all the RNA, you can take all the RNA from a sample, from a cell, you can run it on a gel, transfer it to a membrane, and then you can use a probe to your gene of interest. So if you want to see if the interferon alpha gene is being expressed in your sample, then you can specifically look for interferon alpha. Is it being expressed? And here it is. So you can use a probe. And this is quite an old-fashioned technology. It's useful for one gene. If you want to look at one gene, is it expressed? It doesn't tell you how many genes and their level of intensity too much. Okay. So if we want to look at multiple genes together, we can use microarrays or RNA sequencing. And a microarray technology is a really interesting one. In a microarray, what you have is, you can think of these as little wells or little dots on a slide. With each dot contains a human gene. So this might be the interferon alpha gene. This might be NF-kappa B gene. This might be IL-1 gene, the gene that encodes IL-1. They're all here, okay? And they're made by either PCR or synthesis, and they're placed by a robot into each of these wells. So we've got our DNA from the gene. What are we going to do about it? Well, what we want to do is we want to use this to tell us which genes are actually being expressed in a sample. And in a typical experiment, and this is the example that I want to go through the next 10 minutes, is taking, for instance, a cancer cell. So you can take a tumor from a patient, you take all of the RNA out of that tumor, you then use the enzyme reverse transcriptase to make DNA, okay? And that, so this is DNA that represents all the pieces of RNA in the cell, okay? So basically you're looking at all the RNA transcripts. While you're doing this, you can actually label all of this with a red dye. So in this example, all of the RNA effectively from the cancer, from the tumor, is labeled red, whereas all of the RNA from the, you could take a biopsy of normal tissue from the patient, all of the normal tissue is labeled green, okay? Then what you do is you use this to, you put this onto your slide, okay? And if a piece of RNA, if you like, this is the cDNA that it corresponds to the RNA, if this binds to one of these wells, it means that this encodes, say, if this is a well encoding IL-1, then this is the IL-1 gene has been expressed. Okay, it binds to that, it binds to the particular well of interest. Okay, and this gives you a map like this, which is either red or green or yellow. Okay, so what does this all mean? Well, some of the information is here, so you can see that here, but I've blown this up here. This will be just a portion of that information. So what happens if you see a red dot? Well, say, for instance, maybe this is the NF-kappa B gene, okay, for inflammation, the inflammatory gene. If you see a red dot here, it means that there's more signal coming from the cancer cell. So this means more A than B because it's red. Okay. Here's another gene. It could be some other gene. It's green. What this means, it's more green. You have more green signal, which is from the normal cell. So that is more B than A. Okay. So you can say that this gene is expressed in the cancer cell and not expressed in the normal cell. This gene is expressed in the normal cell and not in the cancer cell. Well, what about the yellow ones? Most of them are actually yellow. And because red and green mix, you mix those colors, you get yellow. Mixture of the colors means that you have equal levels of both A and B. Okay. And what you can actually do is you can develop this chromographic range where red is very highly expressed in the tumor, green is highly expressed in the normal cells, and you have little bits in between. So the red spots indicate genes that are upregulated in the cancer. The green are downregulated in the cancer. Okay. So you can immediately see really interesting technology, lots of data about what genes are expressed in a cell. And and microarrays are still used, but actually they're being overtaken now by RNA sequencing. So I wanted to just highlight the fact, because if you were to go on and do a PhD, for instance, in a lab, you might start seeing that RNA sequencing is used much more. Okay. And in RNA sequencing, what happens? Well, again, here is all your RNA from a cell, from a sample. Chop it all up. Oh, sorry, you make cDNA, okay, using reverse transcriptase. We use cDNA because it's just more stable. It's less prone to break down. But you can think of the cDNA as being representative of all the RNA in the sample. Okay, make the cDNA, then you chop it all up, and then you can sequence all of that. So just sequence it all, and you can sequence this. And basically, what I want you to take on, there's quite a bit of writing on the slide. You can ignore all these junction reads and so on. The main take-home message is that the count data represents the number of times a particular transcript has been sequenced. In other words, if a gene is highly expressed in a sample, you're going to get lots of transcript, and it's going to be sequenced a lot of times. Okay, so in that sample, say for NF-kappa B, that transcript in the cancer cell was very high, then you're going to get lots of reads. You're going to get 8, 9, 10, up to hundreds of reads. So in other words, that RNA is going to be overrepresented in the data set. So the higher the count, the greater the degree of expression. Genes that aren't expressed very highly will give you lower reads because there's not that much of them present in the sample. Okay. This is obviously going to give you a huge amount of data, and I'm not expecting anybody to read that, or you don't want to read, nobody wants to read it, but it gives you a lot of data about which genes are being expressed in the sample. Okay, check my time, and it's all crunched down by a computer. Okay. So both microarrays and RNA sequencing are telling us about the genes that are being expressed in a system. Not the genes that are present, the ones that are being expressed, what's being turned on. And this is very useful. And what I'm doing is I'm taking a very simple example to continue the example of cancer. So the patient has cancer. You can take the tumor from the patient, and you can analyze what genes are being expressed in the tumor. Compared to normal tissue, you can take normal tissue from next to the tumor compared to that. So if this is breast cancer, for instance, a woman with breast cancer, we know that about 30% of these overexpress human epidermal growth factor receptor, HER2. So we can do expression profiling. And if expression profiling shows increased HER2 in the cancer, then we can actually direct that patient to a specific therapy, which is a monoclonal antibody against the receptor, and it's called Herceptin. This is employed, and this is a very effective treatment. But it's not useful. It's not useful unless that patient is expressing the receptor. It won't work. Okay. So profiling identifies the exact repertoire of genes and proteins that are deregulated and identifies susceptibility to different agents. Okay. And so, you know, labs in the United States and elsewhere, they're doing this already. Personalized medicine on a patient, see what genes are being expressed in their tumor, and then directing them to drugs that will potentially work. This is just a very simple example, but there are lots of other examples where you can see, is a tumor going to be drug resistant, for instance? We know that from looking at the gene expression profiling of the tumor. Okay, so I hope you see how useful this information is in pharmacy going forward, and also in biotechnology. We'll get to that a bit more at the end. Okay, so just for completeness, the very last piece of the puzzle is proteomics. So for proteomics, we can use, this is looking at the proteins that are produced in a sample. So you might say, well, maybe this is the most important thing. And we can look at these proteins. There are old techniques that are using gels to run proteins. These are fine. You can only run one protein at a time. But what if you want to sequence and understand all the proteins in a cell, for instance? And this can be done now. And it's done on mass spectrometry. I won't go into the precise details. You know, you might do a lot more on this in chemistry. But what you would typically take is a sample. You'd extract all the proteins. You'd run the proteins on a gel like this. You can run them on a gel. Each line is a different protein. And in the old days, you'd have to cut some of that out of a gel because you might focus on proteins of a specific size. Nowadays, you can actually run all of these proteins. We have the technology to run them all. Take all these proteins out, excise them. You can subject them to digestion, trypsinization, get a peptide mixture. You run this in peptide chromatography and then run it on a mass spec. Okay. And the first run of the mass spec will determine the size, the overall size of the peptides. So what's the overall mass of the peptides? The second one is mass spec, which subjects the system to ionization, breaks down the proteins into individual amino acids, which can then be detected on the mass spec. Okay, so you're getting more and more information as you go through the mass, overall mass of the peptide, and then the actual sequence of the peptide. So you can identify all of the amino acids runs in a sequence, okay? And then you need a computer to work to identify the protein. It compares these sequences to the databases, and it tells you what the proteins are, okay? Because it can identify the proteins based on the sequence, okay? So I haven't gone into all the detail here, but it's just a very useful technique to tell all the proteins in a sample, okay? So what we covered in this lecture are all the different techniques that are used to either determine the genomics, so the gene sequence, gene sequencing, so what's in the DNA, what is being expressed in a sample, using microarrays as a good example, or RNA sequencing is replacing microarrays based on the cheap sequencing technology, and then mass spec proteomics at the end of the day. And you'll be pleased to hear maybe that this is the very final slide. And this is what I want to get across as the concept of systems biology. So we talked about, you know, a real explosion in the equipment and the techniques that are used to generate information about biological systems, okay? DNA sequencing is very rapid, and we can sequence genomes in a couple of days now, human genomes very quickly. But what if you combine this with transcriptome information? What's the cell expressing? What genes are being switched on? Proteomics, what are being made into proteins? And even metabolomics, which I didn't cover, which is the chemistry. So of lipids, carbohydrates, hormones that might be in a system, non-proteomic, okay? Well, the science of this is called systems biology, and it's the science of putting all this information together and making sense of it. And we need bioinformaticians, we need computer scientists to do all this, to tell us, well, which messenger RNA is associated with the disease state. What does all this mean? And this, we're getting towards the science of personalized medicine, where we can take a sample from a patient and say, well, yeah, they're going to respond in a certain way to a drug, and we can use this. We're probably not too far from using that, this information on a rapid scale, okay? Probably maybe 10 or 15, 20 years away, even though it's an expensive technology. As I said, it's already being used in cancer. So we can use big data analysis and statistics, systems biology approaches to compare all these data sets and to say, well, one is up, another is down, this is up, this is down, and so on. It's huge, huge amount of information, okay? So what I'll do is I will, oops, that was the last slide, just checking the chat. And I realize that there's a lot involved in this lecture, but I'm hoping that with the clarity of the slides, you'll be able to go back and understand the basic science of the different techniques, because, you know, we don't have to understand the full science of this. We just need to know how it can be used, and importantly, to look at the examples of why is it important? Why is it important to understand single nucleotide polymorphisms, and where is all this technology going to lead us in the future? Okay. Okay. So if there's anything, you could put it in the chat. If not, thank you for your attention. I can let you go to your next lecture. So shukran, and you can go to your next lecture or wherever you need to be next. Okay, I'll stop sharing my screen, and I just end the meeting. So goodbye, everyone. Good luck in your exams, etc. Thank you. Thanks, Mohammed. Thanks, Khaled. Ahmed. Thank you.